All right, in this video we're going to talk about the very, very subtle and complex, dense topic of polarity. This is based on the overall distribution of electrons throughout a molecule, so it's not a real straightforward idea. The idea is, the basic concept here is that a molecule, molecule, not molecular, molecule with an even distribution of electron density should be non-polar. Okay, a molecule with an uneven distribution of electron density should be polar. Okay, now this is based a lot on the Lewis structure, the three-dimensional geometry, like so the VSEPR theory, the molecular geometry, and it's also based on electronegativity differences. We saw in the previous chapter that if you have two atoms that are exactly the same, these electrons are shared evenly. If you have two things with a difference in electronegativity, the one that has a higher electronegativity, oxygen is closer to fluorine, so it should have a higher electronegativity, are not going to be shared evenly, and the oxygens, the more electronegative one is going to pull harder on these electrons, and you'll end up with a partial negative and a partial positive. So honestly, what we're mostly looking at is how symmetrically the electrons are distributed. Okay, so this is one way to think through it. This is probably the most technical definition. Polar basically means asymmetrical, and non-polar means that it's completely symmetrical. Okay, so the distribution of electrons. So let's just look at a couple examples and I'll try to show you what I mean by this. And then I'll try to explain why we care about this here in a little bit. Okay, so let's look at some really standard examples. Water. Water is pretty common throughout all of chemistry, and so its polarity actually affects a whole lot of things with regards to how Chem 1 and Chem 2, how things dissolve, in organic chemistry, how things react, stuff like that. Okay, so water, we would typically draw it like this. And so if you look at it, what we're looking at is it's an even distribution of electrons. It looks like, okay, everything's symmetrical there, everything's symmetrical there. We said symmetrical means non-polar. The problem is the Lewis structure is not the three-dimensional structure. So we have to use the molecular geometry to kind of figure this out. And I do not absolutely need you to draw things with the correct molecular geometry, but I absolutely need you to be able to visualize it as such. So this molecule is bent. I'm going to kind of draw it like this because that's easiest. And what we're looking for is how evenly the electrons are being distributed. Okay, so we're going to look at this polar and non-polar thing, or sorry, the even distribution or uneven distribution of electrons for electronegativities. So between hydrogen and oxygen, hydrogen is farther away from, dig up a periodic table here. Fluorine is the most electronegative atom on the periodic table. Oxygen is right next to it, so it's very electronegative. Hydrogen is very positive, it's very far away. So what we would expect is that the oxygen will be fairly negative and this will be fairly positive. If you have if you have lone pairs on central atoms, you can almost just visualize those as negatives because they're electrons, they are negative. So what we look at is how symmetrical it is. So if we cut things this way, this side looks the same as this side. If we cut things in the plane of the paper, forward and back look the same. But if we cut things this way, there is a positive side and a negative side, which means this molecule is very definitely polar. Okay, so there's a plane of asymmetry. That's generally how it's thought of. Symmetry plays a critically important part in how molecules work. If you make it to organic chemistry or make it even farther into like upper level inorganic chemistry, symmetry becomes critically important. Okay, but the idea is this molecule is asymmetrical. There is a positive side and a negative side. So the way that we represent this is kind of like a vector, drawing from the positive side to the negative side. So basically we have, this is where the center of the positive is. We basically put a plus sign there, and then that's the basis of our arrow, and we point towards the negative side. I didn't put it directly in the middle because I can't draw. But the idea is this is called a dipole moment. It doesn't have to be red, but I've already started down that path. So this is a dipole moment. And it has to do with how unevenly the charges in the electrons are distributed. Essentially, there's a whole bunch of electrons on this side and way less electrons on this side. So this molecule is going to be polar. Okay. If we compare this to something like CO2, another easy, straightforward example. Carbon dioxide, if you go through all the legwork, looks like this. Okay. And we literally talked about this example up here. We'd expect carbons to be positive and oxygens to be negative. So we do have, sorry, I guess I switched colors here, my bad. We do have an uneven distribution within each bond, but overall the molecule is symmetrical. So if you cut it this way, it's symmetrical. If you cut it this way, it's symmetrical. And if you cut it in the plane of the paper, it's symmetrical. So what we find is that there is no plane of asymmetry, so this molecule should be non-polar. Okay? This is kind of the gist of how this works. Let's try another example, or two or three. Okay. Let's try CH4 versus CH2Cl2. Okay. CH4, essentially what you have is a carbon with 4 hydrogens, and we'd expect a tetrahedral, so I'm going to go ahead and draw it as a tetrahedral. Again, I don't need you to be able to draw the wedges and dashes, but I do need you to be able to visualize it correctly. Okay. This is going to have 4 groups attached as well. They're going to be evenly distributed. What I need is for you to think through the fact that this is still a tetrahedral. Now, where we put everything doesn't really matter. I'm going to put the 2 hydrogens over here and the 2 chlorines over here. If I had chosen different options, if I'd put the 2 chlorines here, the 2 hydrogens here, or the 2 chlorines here and the 2 hydrogens there, you'd still get the same answer. I'm going to put the lone pairs on here because these are technically Lewis structures. And what we find is that carbon and hydrogen actually have almost exactly the same electronegativity. So the carbons are very, very slightly negative. I'm going to draw these super small. And this is very slightly positive. It is a very minor difference. Say, so same thing here. What we're doing is we're comparing everything to the center. So honestly, a lot of times we don't put the negative or positive on the center. But compared to the center, hydrogen is slightly positive. But chlorine, in terms of electronegativity differences, chlorine is right next to fluorine and carbon's pretty far away. So there is a very definite negative on the chlorines. So if you cut this in the plane of the paper, everything's symmetrical. If you cut it like that, we have a negative side and a positive side. So So this molecule would be non-polar, and this molecule would be polar. Okay. So here's the underlying problem with all of this. This is not how it works. This is how it works, but it's not as simple as we're making it. And unfortunately, a lot of textbooks and standardized exams and labs, they'll say, is it polar or non-polar? That's not really how it works. The way it really works is something can be completely non-polar. If there is a completely even distribution of electrons, like this is completely symmetrical in all directions, it's non-polar. If you have a complete separation of charge, like one side has like a bunch of full negative charge over here and full positive charges over here, that molecule is ionic. Anything between here and here is polar. So if you say, is something polar or non-polar, that's like being given a black and white. Like, it's like, you know, suppose this is white and this is black, and you're given something that's gray, and then you're asked, is it black or white? It's gray. It's literally the gray area in between. So the idea is, what I will usually do on a quiz or exam is I'll give you two molecules and say which one is more polar. Honestly, CH2Cl2 is a very, very non-polar molecule. It's technically polar, but it's like, if you had to plot it on this graph, it'd be about here. So, I mean, I guess of the molecules we've done, CO2 would probably be over here, CH4 would probably be over here, CH2Cl2 would be about here, and water would be way the hell over here. Okay, so in all honesty, CH2Cl2 is more similar to these ones than to water, even though they're both categorized as polar. So what I would do is I would give you two molecules that are similar, like both of these are tetrahedral. You can compare them directly easily, and I'll ask you which one is more polar, because that is usually a more relevant question. Okay. So, let's try to talk about why we care about any of this. Okay. Why would we care about polar molecules or non-polar molecules? What I will tell you is that polar molecules dissolve in polar solvents, and non-polar molecules dissolve in non-polar solvents. And even that is an oversimplification. But the idea is that polar and non-polar don't mix. Okay, the stereotypical example that everybody always uses is Italian dressing. Okay, if you have oil and water, they don't mix. Oil tends to be very, very non-polar, so example, Italian dressing. Can't spell apparently. Italian dressing. For anybody who doesn't know what Italian dressing is, it's typically the bottles come in this shape at the store, little screw top lid, whatever. What it is, if I remember correctly, it is vinegar and olive oil with a bunch of spices in it. And what you end up with is two layers. This is the oil layer, which is non-polar. And this is the vinegar layer, which is basically dissolved in water. So vinegar in water, which is polar. And then there's various spices and stuff. But the idea is that basically these things will never mix, which is why anytime anybody gets Italian dressing, they usually shake it up a whole lot until you get a bunch of bubbles and everything's as dissolved or mixed as it's gonna get. And you pour it on your salad real fast. And then what I will tell you, I'm not a big fan of this, I'm gonna make fun of Italian dressing for a minute, is that it's immediately going to separate again. And the bottom of your salad is gonna be wet and vinegary, and the top is gonna be oily and slimy. I think it's a bad plan. I don't recommend it, but this is an example of polar versus non-polar. A better example, a more relevant example in the real world, is the idea of micelles. Any of you bio people hopefully know how this works. If you have fatty acids, they're kind of weird molecules. They are long carbon chains with usually some sort of polar end. So they're actually both polar and non-polar. So they're molecules that have a big chunk that is polar, or sorry, this part is non-polar, probably should get this right. Non-polar, and then they have a polar end. And what happens is if you lump these things together, polar sticks to nonpolar and nonpolar sticks to nonpolar. So polar and polar work together, nonpolar and nonpolar work together. So what happens is if you have a bunch of these laying around, I'm going to just kind of draw them as zigzags, the ends of these things are all going to end up together. And the tail of these things, they kind of look like sperm, sorry, kind of work like this. And then on the other side, you can also have more of these. And then the whole thing will form a ring. Well, actually it forms a sphere, but we're doing it 2D here. And the idea is you can have water on the outside because water will interact with the polar end. So this part is polar, and this part is polar, and you'll have water in the middle. The inside part is non-polar. And this is how your cells work. All the cells in your body basically have fatty acids, like forming layers of polar and non-polar to separate the different parts of your body. This is how drugs work. There are certain medications that work better as polar or work better as non-polar. Polar and non-polar dictate everything in biology. It's a hugely important concept. So we're not going to go too far down this rabbit hole, but please understand that this is really important. For quizzes and exams, mostly what I'm going to ask of you is I'm going to give you two molecules that are pretty similar and ask you which one is more polar. So what I'm going to ask you to do is something like this. Draw both of them, be able to visualize what they look like in 3D, and just see where the positives and negative parts are. If they're evenly distributed, they're non-polar. If they're not, they're polar. A slight aside here, a slight little fine print. If you have a molecule that's pretty much only carbons and hydrogens, so however many X's and however many C's and H's you have, they're almost always non-polar. So honestly, like I went through all the legwork on this one, but I just saw C's and H's. The answer is non-polar, and you don't really have to think that hard about it. Okay. So when we talk about whether or not things will dissolve in water, this is what we're talking about. Water is polar, so something that is non-polar will not. Okay. All right. Thanks.